Hey everyone, how are you? Did you guys enjoy yourself here at UCLA? Right on, go Bruins. A little backstory, and I'm going to try to rush through some of the more laborious slides so that you guys can relax and maybe incorporate some of the main findings. I was a tech here back in the late 1990s. I studied pediatric brain tumors for a long time. We started studying these things called cancer stem cells and brain tumors. It was fascinating. It was remarkable. It really, I found a future and a purpose in it. And then I did my Ph.D. here in molecular medical pharmacology. So I'm a pharmacologist and drug developer. I did my postdoc in bioengineering because developing drugs, and especially cancer drugs, are very difficult. So I took some extra training in bioengineering. Did I lose my talk? Am I hitting the right button? Up. Up, up, up. All right. All right, so the malignant brain tumor. Glioblastoma brain cancer is a largely untreatable deadly cancer. Median survival ranges from around 12 and a half months to 16 and a half months. Disease is characterized by extreme cellular and molecular genetic heterogeneity. The standard treatment involves surgery, radiation, and chemotherapy. It is the first soft tissue cancer to be shown to be a disease of cancer stem cell origin, and there's really no predisposing factors. The cancers happen, and we just don't know exactly who or when it's going to happen. But glioblastomas have a tendency to be related to older age, but we also find glioblastomas in pediatric populations as well. Brief history of cancer stem cell biology. This is about 15 years in one slide. Cancer stem cells are essentially highly malignant supercells. They hijack natural stem cell survival and cell proliferation processes. They self-renew to replicate themselves. That's very important in stem cell biology. And then they create the cells which produce the tumor bulk and which patients ultimately succumb. In 2003, cancer stem cells were first isolated in vitro and expanded from pediatric brain tumors like I was describing. And in 2013, 10 years later, which sort of describes how long it takes once you go from a discovery to application. In 2013, cannabinoid receptor agonists, both CB1 and CB2, were found to be potentially effective in killing glioblastoma cancer stem cells. This was drug screening we did, and it was directly on these cell populations, and it was a way for us to see, oh, we can use these types of cell models to study drug development, and we can also find insight into what drug classes might potentially be used therapeutically for glioblastoma. And in the last 15 years, which this point relates to the fact that the cancer stem cells aren't just in brain tumors. They're in a number of different cancers, and the same models have been used repeatedly with regards to the culturing processes of taking cancers, putting them in a petri dish, looking for stem cells on it, and it's called the sphere system, which you can see at the bottom. Those are balls of cells, and that's how stem cells look like in a petri dish, except these are cancer stem cells. So in the last 15 years, cancer stem cells have been identified in a number of malignant and metastatic cancers: breast, prostate, ovarian, colorectal, pancreatic, melanoma, and some others as well. Oh, you know what, I want to back up. On the right, the pretty picture of orange and green and whatnot, that is when we try to differentiate these cancer stem cell spheres to see whether they can turn into other cell types. And so the green labeling is for a marker for neurons, and the orange labeling was actually red, but there's red and green, and they make orange. And those are, the red markers were markers for neural progenitors. And those are some of the earliest studies that we were doing and trying to see what we actually had in a petri dish and study this cancer stem cell phenomenon. The science behind cannabinoid-based cancer therapy is rooted in signaling pathways. If you walk away from this meeting and somebody asks you at the dinner table, what is this deal about cannabis potentially being used as a treatment for cancer? It's based in signaling pathways that we've been studying for a very long time. They're very complicated to develop drugs for. I'm going to just zero in to what we need to be focused on in this slide, is that in the very middle you're going to see a red and green. That's AKT and mTOR. mTOR and AKT are considered sort of like a therapeutic graveyard, if you will. We try to develop these drugs. They don't get to clinical. If we're trying to target AKT and mTOR, very difficult drugs to develop. A lot of the drugs that we try to move through the clinic don't make it. They fail in preclinical. So this is going to become very important because I'm going to talk about AKT and mTOR, and you're going to walk away with AKT and mTOR like, oh my God, this is the thing. These are the things that we're going to be using in the future to treat cancer. The bottom of the slide is all the cancer things that go on in a cell regarding cancer. So on the right side, epithelial mesenchymal transition, called EMT transition. That's a mechanism by which a cell can pop off from a tumor and then go somewhere else. So that's a mechanism of metastases. It's a stem cell process. Tumor progression and angiogenesis, cell death, autophagy, apoptosis. That's all we're looking for when we try to develop drugs is, can we kill the cell through apoptosis, which is cell popping, and autophagy is a realignment of the cell that produces cell death. Cell proliferation, obviously very, very important in tumor biology. And then cell cycle, we try to block the cell cycle. The green arrows are activating signals, and the orange button is an inhibitory signal. So there's a lot of activation and inhibition going on in a cancer cell, and a lot of this happens in cancer stem cells. The little pink thing up top is EGFR. That's going to become important to some of the data. That's Epidermal Growth Factor Receptor. It's sort of like HER2, but a different variant of that. But it was very important in terms of the data I'm about to show, which is single-cell heterogeneity analysis of glioblastoma cancer stem cells. There's a lot of, in order for us to use this as a procedure or way to identify what works, what doesn't, and develop therapies, we had to have some decent, better tools. Because most of cell biology is done. We use a lot of different cells, and then we'll crack open like 1 million cells and then analyze them. And single cell analysis basically is a lot more high content. It's quantitative, and it allows us to like label the cells, identify signals, incorporate other signals that might be going on in parallel or in concert with. And we use these microfluidic chips to so on the top slide is glioblastoma. We take the glioblastoma, we dissociate the cells, put them in a petri dish, spheres form, then we break up those spheres, and then we put them in these little microfluidic chips, and this allows us to do really high sensitive, low cell number analyses. The microfluidic chips were developed at the California Nanosystems Institute, just about a block and a half away from here. We're looking at EGF. We were looking at the relationship between EGFR and tumor suppressor P10, phospho-AKT, and mTOR. And what happens is we overlay the cells. It's basically quantitative immunocytochemistry. We can quantitate the signal based on the amount of protein that is fluorescently labeled. So the colors represent the fluorescent labels. And we, as I said, we looked at EGFR, P10, phospho-AKT. And then the third thing is validation, which is a way to quantitate and see, like, the signals we find, are they relevant? Can we make use of it? Experimental data set, 15 samples, 4 biomarkers, 1,000 cells a sample, 60,000 data points, 15,000 cells is all the cells I needed to do the analysis that I'm about to show you. More quantitative analysis. Algorithms basically derive the relationships within and between cells and patients. We use self-organizing maps. They'll take a molecular signature in the data set, the markers quantified, cell percentages are mapped. This is what these signatures look like. 15 samples, and their molecular signatures, and each little circle represents a one unique molecular signature. So brain cancer stem cells have essentially up to 49 molecular signatures, and this just sort of organizes that, and it's according to the patient sample. So so we're able to estimate heterogeneity. From that mapping, we do a thing called neighborhood frequency vectors, and then we do unsupervised clustering. This is the thing that just excited me. It's excited me for years and years and years now that we're finally getting ready to, we now have some good data set, enough understanding of this data to make sense of it and publish it. And on the left, there's a unsupervised hierarchy clustering. Each column represents one patient. Each row represents a unique molecular, one of those 49 molecular phenotypes. To walk away from this, because the time started going down, molecular signatures are defined by differences in EGFR expression, which is in pink, and activation of AKT in red, and mTOR in green. And there's a little cheat sheet on the lower left-hand corner. Like I was saying, that's the therapeutic, those are the places that we have very difficult time to develop drugs. The little black circle, AKT and mTOR is right in the middle of that. Incredibly important for developing therapies for cancer and cancer stem cells. We did some validation measures real quick. We just basically grew spheres and then counted the spheres and found out whether there was a malignancy associated with the cluster 1 and cluster 2. Cluster 1 had a higher malignancy signature because we needed less cells to plate to form these little spheres. And then cluster 1, we needed more cells to get an analysis of saying, how these spheres grow. And it was just, lots of spheres come from highly malignant brain cancer stem cells. Not a lot of spheres come from cells that are not very malignant, and so you need more cells to put in a petri dish in order to do your studies. And so that's what that represents. AKT mTOR activated cluster 2 had significantly higher sphere formation and had higher sphere forming efficiency as an indicator of malignancy. We looked at patient outcome because we had the data on the patient. While we were working in the lab, these patients would eventually succumb to disease, so we had outcome measures of progression and survival. And these things act as curves, and the curving is, if the curve goes farther along the x-axis, the patient is doing all right, they're surviving longer, or they have less, they're not progressing, or the MRI doesn't show a recurrence or anything. If the x-axis is closer up to the origin, the patient doesn't do as well. And we use these in terms of identifying what molecular signatures or what we're supposed to be looking at might be related specifically to patient survival as an outcome measure. And it's very important in drug development, is having effective outcome measures. Targeted cannabinoid therapy. Cannabinoid receptor agonists may target cancer stem cells by directly inhibiting PI3 kinase, AKT, mTOR or TORC1, which is a TOR complex 1, and other hard-to-target cancer signaling pathways. I'm not going to go into this in a lot, but basically it goes right back to the fact that these are very difficult proteins to target. They are part of the cancer stem cell machinery, and I'm going to show some clinical hope and promise. Now, I had to skip straight from basic science right into clinical hope and promise because this is really exciting. This is the thing that I was able to look back at my data and say, oh, we have something here, that the story makes sense. In early 2017, GW Pharmaceuticals found positive results in phase 2 proof of concept study in glioma, recurrent glioblastoma, 21 patients. They used a THC CBD formulation. They had a 1-year survival of 83% using the THC CBD formulation versus 53% placebo or no formulation at all. And then their median survival was 18.3 months with on the formulation versus 12.3 months placebo. In most universes, that would be considered blockbuster or approaching the direction of blockbuster. So, this is really, really exciting things. It's very good for the field. It's very good for our understanding of cancer biology, what we're going to be looking at in terms of treating cancer, how we diagnose things a little bit better so that we can apply the appropriate therapy. If it's a cannabinoid therapy or it's some other signal transduction pathway blocker, we've got to sort these things out. The closer we get to the exact signaling pathway of what a patient has, the better chance that we can do better clinical trials and directly target the right therapy for the exact patient with the exact sort of mutations that they might have. Coming from there, the press release from GW Pharma, cannabinoids have been shown to promote autophagy, which is the process of, and I always mispronounce that, the process of regulated self-degradation by cells via several distinct mechanisms, including acting on the AKT-mTOR pathway, which is an important intracellular signaling pathway that is overactive in many cancers. So it's basically a convergence. I don't work for GW Pharma, didn't really know about what they were trying to accomplish, but our data, it converged with their data. And perhaps the future might be, yes, we're now looking at cancer stem cells. These are brain cancer stem cells we're looking for. They're in the patient tumor. The cannabinoids are targeting these specific cells, and this is a potential improved way to look at treatment, be it as an adjunctive or as a full-on therapy. But we now have the science there. We now have the potential of these botanical drugs there, and this is all very promising. And when multiple groups come up with the same solution, that's the sweet stuff. That's what we need to be doing. All right, cures come through collaboration. I feel like I'm getting choked up. It's been a long time since I left UCLA like 5 years ago, and I was like, I gotta go do my own thing now, and so it's great to be back. In the cancer stem cell biology crew, Professor Harley Kornblum really stewarded all of the brain cancer stem cell work. He was a mentor of mine. Professor Ed Panosian, he's a pediatric oncologist here down in Harbor. Professor Medeo Cho was a resident in the lab, did a lot of basic science studies that we're working on. She is now in the brain injury program in the Department of Neurosurgery at UCLA. A lot of other, Copan, Tiffany, Jack, Eric, these are my best friends. We worked very hard on very difficult problems, and I love the way they supported the work that was going on. Microfluidics, Professor Jing Sun is a professor in China now. She was my postdoc when I was a grad student. Professor Ken Kamei, he's now doing stem cell work down in Japan, biotechnology and stem cells in Japan. Minoru Hase, she is going to be an MD PhD candidate here in the School of Medicine. Dr. Bill Wong, pathology, taught me every single thing I knew about pathology, and was just an incredible source of support. In neurosurgery, Professor Jorge Lazarev, he is a pediatric neurosurgeon that basically green-lighted all these studies to begin with, and went on to do he's a professor emeritus now. He was a close mentor to mine, along with Harley Kornblum, and Linda Liao they're the two neurosurgeons. Jorge and Linda had done excellent work in making sure that we got the samples we needed, and these are all through extensive collaborative efforts. Linda is now the chair of neurosurgery here at UCLA, and then in bioinformatics we had another great team, Tom Graber and Nicholas Graham. They really just came through, and we had all this random cell data, and they really made good sense of it. And Tom is in CNSI, and Nick Graham went on to USC. So I want to thank you, CanMed. Go Bruins. We're all in this together, and I think we've got a great future with cannabis-based medicine for going after some really difficult and hard to treat diseases. I really wanted to make time for questions, but I got like a couple minutes here. So if anybody has a real question, come up to the mic real quick so we can get it in. I'm going to talk about, this is in memoriam to our lost Mavericks. Senator John McCain died of a brain tumor. This picture was taken, if you can look over, see his left eye or right eye? Is that left eye? Left eye. He had just come out of surgery a few days before this picture, and he was back in Congress doing what he needed to do, and he died last month. Beau Biden, Vice President Biden's son, died in 2015, and then our partner on the agricultural side, we're in the process of trying to, we've got to create this medicine from somewhere, so I worked with agricultural partners, and moving forward. And Josh Yastanko was just an agricultural pioneer, taught me just about everything I need to know about the nature of how we're going to turn this into medicine. And me coming from the lab and then all of a sudden being embraced by an agricultural field was really wonderful. And we lost him to a car accident last year, just out of nowhere. But that's just how it is. So in memoriam to our lost Mavericks, we do this for a living. There's always somebody that we're going to be helping out as we move forward. So all of us that run into all these catch 420s, get used to it, but you can do great work with it.
We have 1 minute.
Thank you. My name is Kristen Wolschlegel. I'm an RN, or oncology nurse navigator, doing this work for about 2 years exclusively with cancer patients using cannabis, as well as integrated into conventional care. I see a trend that I haven't heard talked about in research recently. Neuroendocrine tumors seem to be very vulnerable to some combination of cannabinoids, whether it's neuroendocrine tumors of pancreas, cervix, small cell lung cancer, metastatic prostate cancer, and I just wanted to raise awareness because it sure seems to fit in the category of stuff you're interested in looking at.
Yeah, I'd love to talk.
Neuroendocrine differentiated tumors.
Yeah, I'd love to talk to you and follow up about this, because these are the kinds of things where we have these conversations and then we have to go through what is the real clinical process that we would take to do a clinical trial. And if we start talking, then we might be able to put a stitch in time with regards to these clinical trials and get these therapies to out to the fields and into the doctor's hands quicker. Thank you.
Okay, thank you. Round of applause, please. Michael Mastermann Smith. Next.